Hello friends, warm welcome to you all to the channel World of Zoology. This is our second lecture on the unit Instrumentation from the paper second, and this lecture is on microscopy. We'll start with the same in this lecture with the description of dissecting microscope and compound microscope in today's lecture. The points to be considered in this lecture are construction, then principle and application of dissecting and compound microscope. First, we'll consider the first point, that is introduction. A cell is a fundamental structural and functional unit of life. The body structure of all living organisms, including Monera, Protista, fungi, plantae, animalia, are a form of cells which are invisible to the human eye. To see and to study the cells, a tool, that is instrument called as microscopy, has to be used. Advancement made in the studies for understanding various living forms from bacteria to protozoa to larger creatures, that is multicellular animals, and the subcellular aspects of the individual cells have simultaneously led to the development in the microscopy. Microscope, the term is consist of two small terms. First, that is micros means small and scopy means to view, is a seer of small things, that is which can observe all small things. Robert Hooke was one of the first to use the microscope to describe structure of living material. When looking a thin slice of cork, he saw little boxes arranged like honeycomb. He coined the term cell to describe each tiny individual structure, as it reminded him the stark living quarter of a monk. It is possible to enlarge and even resolve the image of an object by using a system of lens or lenses and sources of light to illuminate the object. In simple microscope, like magnifying glass, single lens and visible light is used, whereas in compound microscope, more than one convex lenses are used. The source of light can range from daylight to light in visible range of the electromagnetic spectrum to a beam of electrons or photons. In simple microscope, light from source passes through a convex lens to get an enlarged image of an object, which is viewed from the another side or another end. The lens magnifies, that is, enlarges the object, depending upon the focal length of the convex lens which is used. A wide range of microscopes are available to the modern biologists, from a simple microscope such as magnifying glass to dissecting and compound microscopes that use more than one lens to magnify and resolve an image. Microscopic structures that are difficult to visualize under compound microscope are visualized using advanced sophisticated instruments such as the scanning electron microscopes (SEM) and transmission electron microscopes, that is TEM. The SEM and TEM utilize the electron beam and electrostatic and electromagnetic lenses for visualizing the surface and cell organelles of the micro dimensions. Ray diagram for a microscope where U and D being the distances of the object and its image respectively, that is U distance of the object and distance of the image is represented by capital D, letter D. AB and A1B1 being the size of the object and image respectively. Structure of the dissecting microscope with stage, then convex lens, and the adjustment screw and the particular structure on the arm so as to hold the lenses, or we can change the lens with the help of the adjustment and requirement of the purpose or the image to be observed. Construction of a dissecting microscope. A dissecting microscope consists of a stand having base or foot, a stage, and adjustable limb. It has a paired optical lenses fitted into eyepiece and is fitted with mirror which can be rotated at 360 degrees on two axes for reflecting the required intensity of the light. The paired optical lenses that forms the eyepiece can be raised or lowered by means of focusing knob. Some of the advanced dissecting microscopes having a binocular vision with two eyepieces and may have an inbuilt source of light. Magnification of an image depends upon the magnification power of the given lens or lens which are to be used. Magnification is often expressed as the number times of x. For example, the eyepiece may be of the power 10x, which means that the lens can enlarge the image 10 times the size of the object. If it is 20 times of the object. By changing the lens to 15x, the magnification can be increased to 15 times of the object, size of the object. Application of the dissecting microscope. The image is obtained due to reflection of light by the object rather than the light that passes through the object. This arrangement has an advantage in that the user can view opaque and large specimens. Second application, as the eyepieces are removable, they can be replaced with lenses of different magnifying power, thus making it flexible for use or according to our requirement. The eyepieces can be changed. It has more working space between the lens and object on the stage. This makes it easy to manipulate the object under study. Even the dissection of the particular microscopic organism is possible because of that. An object can be illuminated by ordinary daylight, making it possible to use it for fieldwork. It can be used for preparing specimens for examination under higher power. It can facilitate dissections of smaller animals and temporary preparations or mountings which are required during biological study. Here the next part, that is structure of the compound microscope after completion of the dissecting microscope. 3 points of the dissecting microscope: the structure, then principle and application of the dissecting microscope. We'll see the next, that is compound microscope, with the same points. Here the structure is shown of the compound microscope with eyepiece, then the arm is there to which at the upper end the adjustment screws are there, coarse and fine adjustment. Then the lower side contain this particular structure, that is the base of the microscope or foot of the microscope to which the mirror is attached. Then the stage portion is there on which the slide is mounted, and then to the body tube at the one end eyepiece is there, and at the lower end near to the object or the stage of the microscope the lower power objectives are there with revolving nose piece. Low and high power objectives are attached. The construction of compound microscope is similar to that of a dissecting microscope, namely the mechanical system and optical system. The compound microscope has a heavy base with stage fitted with mirror having concave surface on one side and plane surface on the another side. In addition, a condenser with convex lens and a facility to raise or lower the same helps condensing the light rays reflected by mirror before they fall on the object placed on the stage of the microscope. The condenser is also fitted with diaphragm for adjusting intensity of the light rays. Filters of various wavelengths, that is colors, can also be used along with the diaphragm if needed to provide the contrast. A metal cylinder is fitted over mechanical gear system connected with coarse and fine adjustment screw knobs. This cylinder holds the eyepiece at one end and objective lenses at the opposite end for giving clear picture of the object. The mechanical gear system provides movement to the optical cylinder for adjusting focal length with the knob of the core adjustment screw followed by the fine adjustment, that is fine tuning with the knob of the fine adjustments. This enables observation of the magnified yet clearly resolved image of the object. The magnification depends upon the magnification power of the eyepiece and that of objective lens which is used. This example for this, a combination of 10x eyepiece with objective of 40x magnifies the given image to 400 times of the size of the object, that is 10 into 40x. The compound microscope can be fitted with eyepieces with wide range of the magnification such as 10x, 15x, 20x. Similarly, the objectives have magnification power of 4x, 5x, 10x, 40x, 100x, etc. An objective lens of 10x power is referred to as low power, while upper version are referred to as high power objectives, where 100x is also called as the oil immersion lens, since a drop of oil is placed between the objective lens and the cover slip or the slide to manipulate the refractive index, achieving high resolution so as to observe the object clearly. The objective which has the closest proximity to the object being viewed has several major functions. Which are the functions given below? 1. The objective gather light coming from the various points of the specimen which is placed on the stage of the microscope. The objective reconstitute light coming from the specimen into corresponding points in the image. The third function of the objective projects a magnified real image up into the body tube of the microscope to be collected for further magnification by the eyepiece. Eyepiece or ocular, which is fitted with body tube at the upper end, is farthest optical component from the object, having proximity to the eye of the observer, and eyepiece has the following major function. First, the eyepiece serves to further magnify the real image projected by the objective. The second function of the eyepiece, it produces a secondary enlarged image. Third function, the eyepiece can be fitted with scales, markers, or crosshairs, often referred to as graticules reticules, or reticles in such a way that image of the scales can be superimposed on the image of the specimen for the measurement of the particular specimen. An artificial or natural light source of the object for examination under a compound microscope, and hence it is also called as light microscope, as the natural light source of the object is used for the examination. Here it might be noted that the term light microscopy refers to the method by which the image of an object is being transmitted to our eyes. A microscope helps in the study of the object or specimen by the two ways. One, it magnifies or enlarges, and secondly, it resolves the image of the object. It must be remembered that magnification reduces the field of view of the object. The separation between internal parts of the object get diffuse. This ability to distinguish between separation of smaller objects within the object being studied is called as the resolution or resolving power of the microscope. That means this ability to distinguish between separation of smaller object within a particular object which is studied is called as resolving power of the microscope. This power of resolution depends not on the system of the lenses but on the nature of the light used for illuminating the object or to create its photographic image. Objects smaller than wavelength of the electromagnetic radiation used cannot interact with it and so will be invisible. However, seeing or visualization of such an object can be achieved by using a radiation of very high frequency, as may be given by, say, an electron or a high energy photon, which has a wavelength that is small enough to interact with the object. This interaction can make the radiation used to be reflected and create an image to be photographed and seen. Now, regarding the next point, that is applications of the compound microscope. First application, different parts of the animal tissues can be studied in details for understanding their fine structures. Pathologists can use the compound microscope to study the human cells and tissues for disease diagnosis so that treatment may be decided upon the or the develop and therapy is given to the patient. Microbiologists use it to study the bacteria and other microorganisms, and again it can be used for diagnostic purpose also, say for example presence of entamoeba, presence of signatoring stage of the malaria, which is for diagnostic purpose, and that is studied by culturing or preparing smear of the blood and then identifying the particular parasite within the blood or the fecal matter, whatever it may be the sample. Chemists find it useful to study structures of the crystals so that their nature and composition could be understood. Botanists use it to study the plant material, for example, to study fungi and other diseases that may be present on the plant surface so as to decide upon the measures to be taken to protect and cure the particular plant. In forensic science, compound microscope is used to examine the crime evidences or the microscopic structures which are present at the scene of the crime. Thank you. for watching my video on microscopy. Do like if you like the lecture and share the lecture for the benefit of the maximum students. Subscribe the channel World of Zoology for the intimation of the next video on the same topic, instrumentation.